Hello, my name is Kivilcim Ozturk. I work in Carter Lab at UCSD, and the title of my poster is Predicting Functional Consequences of Mutations Using Molecular Interaction Network Features. Variant interpretation remains a central challenge for precision medicine. Missense variants are particularly difficult to understand as they change only a single amino acid in a protein sequence, yet can have large and varied effects on protein activity. Numerous tools have been developed to identify missense variants with putative disease consequences from protein sequence and structure. However, biological function arises through higher order interactions among proteins and molecules within cells. We therefore sought to capture information about the potential of missense mutations to perturb protein interaction networks by integrating protein structure and interaction data. Firstly, we constructed a structure-resolved PPI network, called here SRNet, comprising more than 6,000 proteins and 10,000 PPIs using 3D structures and homology models. We labeled each amino acid position as core, surface, or interface, defined as a subset of amino acid residues that mediate physical contact between proteins. We mapped various somatic mutations and germline variants to network edges to describe their potential to impact biological function and show that disease mutations frequently target interface or core residues. We then designed 16 features describing network-level effects of mutations, 7 protein-level features that estimate the significance of the target protein in the network, and 9 residue-level features quantifying the potential of individual amino acid positions on the protein to impact network architecture, and evaluated them within a machine learning variant classification framework. Network-based features show potential to distinguish between different classes of variants and are not strongly correlated with other classic non-network-based amino acid features used for variant classification, suggesting that they add new and useful information. Then we trained a random forest classifier on cancer missense mutations, designated as likely driver and passenger, using different combination of features and five-fold cross-validation with gene holdout to prevent information leakage and consequent overfitting. We evaluated classifier performance using all 16 network features, only the 7 protein level features or the 9 residue level features, or in combination with 83 classic non-network based amino acid level features. Addition of network-based features to classic amino acid level features significantly boosts performance. Distribution of residue-level network features for correctly labeled driver and passenger interface mutations occurring on the same proteins suggests that they provide information useful for within gene mutation classification. We assessed the performance of our classifier, which uses all network-based and classic amino acid features relative to both cancer-specific and population-based methods. We evaluated performance on 4 external pan-cancer datasets constructed using different approaches, an in vivo screen, an in vitro assay, and 2 literature-derived datasets. Our classifier, named here Network NAA, performed well based on the area under the ROC and precision recall curve scores across 4 benchmark sets relative to other state-of-the-art variant classification methods. So in conclusion, we show that network-derived features can capture novel information about variant effects that is not already present in the classical amino acid features. That combining both sets of features improves classifier performance, and residue-level network features are helpful for distinguishing between variants within proteins. However, it is important to note that our method has a limitation of the coverage of mutations due to the requirement for structure and interface information. In conclusion, our study suggests that information about molecular interaction networks can be incorporated into machine learning-based variant interpretation frameworks. This opens future directions for the development of novel features capturing network information. For complete details about this study, please refer to our paper listed under the references section. Thank you all for listening.